So, where are we headed? So what we're going to do today is we're going to talk about excitation-contraction coupling and the sliding filament theory. And so what we're going to work our way through first is through a couple of slides talking about the neuromuscular synapse and the motor unit. I'll first show a slide for a motor unit, and then I'll go ahead and quickly describe what we've already talked about in terms of acetylcholine release, the excitatory postsynaptic potential, and from the excitatory postsynaptic potential that's generated from the opening of these ligand-gated ionotropic channels will then generate an action potential. Now, this is the same story we've seen so many times before, and the reasons for that action potential are again the same. That is, we need some kind of long-distance signaling mechanism. And that long-distance electrical signaling mechanism, again, is going to be the action potential, and the excitatory postsynaptic potential is going to be the intermediate, and we'll talk a little bit about that. But to understand how the ideas of neurotransmitter release, excitatory postsynaptic potentials, and action potentials are functioning to cause muscle to contract, we have to spend some time on the membrane systems of muscle fibers. And so we'll then emphasize how these muscle cells are really peculiar. They're really, really unusual. They're very long. They can be up to a centimeter or more long. And they still have a plasma membrane called a sarcolemma. But in addition to the sarcolemma, these cells have a transverse tubule system, that is a T-tubule system which delivers action potentials to the sarcoplasmic reticulum. There's going to be a site, a junction, called a TSR junction, where the T-tubule system meets the sarcoplasmic reticulum. That's going to be occupied. This gap is going to be bridged by two very important proteins, one called a dihydropyridine receptor and the other called a ryanodine receptor. And these are peculiar because the dihydropyridine receptor is actually going to form a block, a plug, that fills the ryanodine receptor channel. The ryanodine receptor channel is in the endoplasmic reticulum, or the sarcoplasmic reticulum, whereas the dihydropyridine receptor is found in the T-tubule membrane. These two are going to be intimately related with one another in such a way that when the action potential arrives, the dihydropyridine receptor will pull its plug out of the bottle of the ryanodine receptor, allowing calcium to flow from the sarcoplasmic reticulum, this calcium storage organelle, into the cytoplasm. So that's going to be pretty much the membrane and electrical story. But we now have to go from the electrical membrane story to this idea of excitation-contraction coupling. How do we go from electrical signals to mechanical movement? To do that, we have to understand how the cytoskeleton is organized into this repeating structure called a sarcomere. Every muscle cell has something in the order of 1,000 or more, maybe 10,000 of these sarcomeres laid out in series, one after another after another. And these sarcomeres laid out in sequence then, in series, are organized in a repeating pattern. The repeating pattern is such that what you see is that there are these actin-thin filaments. Now, we've talked about actin-thin filaments before, but we've talked about them in terms of how they're organized in the cytoskeleton. But again, we're going to see a filamentous actin structure. But associated with this filamentous actin structure are also going to be two other important proteins. Troponin, which is organized in something known as a troponin complex. It's in fact a calcium binding protein. And tropomyosin. Tropomyosin, which is actually we're going to see, is a kind of shielding protein. The long and the short of this is the calcium is going to be released from the internal storage compartment. The calcium will bind to the troponin complex, move the tropomyosin, and expose the actin. It's going to expose binding sites in the actin, which can be attached to by the myosin-thick filaments. The myosin-thick filaments, and we've talked about myosin before, but now the myosin-thick filaments are going to be in bundles. They're going to be in bundles, and they're going to look like a bundle of sticks with these heads sticking out. The myosin structure then allows for its heads to reach up and contact the actin. But they can only reach up and contact the actin after calcium is bound to the troponin complex and the tropomyosin is moved out of the way to reveal the actin binding site. And we'll see how that gets done. Well, the third character in the play is going to be the alpha-actinin. This is an actin binding protein, but the alpha-actinin is organized in this repeating pattern such that it forms the Z lines. This will be the boundaries, the endpoints on each one of the sarcomeres. So at the ends of the sarcomeres, we're going to have the Z lines. The actin will be bound to the Z lines. These alpha-actinin Z lines are in turn tied into the membrane. So now we have a mechanical connection between the cytoskeleton and the plasma membrane. And then finally we'll talk about the actomyosin bridge formation, how the myosin head reaches up, contacts the actin, undergoes a power stroke, pulling on the actin, tugging on the Z line, drawing those Z lines closer together. In a typical muscle contraction, the distance from one Z line to the next typically a typical distance of reduction from one Z line to the next is about 20%. So you'll see that the muscle will go from, say, 1 millimeter down to about 0.8 millimeters. But what this means then is each one of these sarcomeres laid out in sequence, each one shortens by about 20%, so that in aggregate, in composite, when you have all of these laid out in series, you get a total 20% reduction in the length of the muscle fiber, and that's how we shorten the muscle fibers. And we'll talk about how that gets done.